we are going to talk about other methods like budding, fragmentation and so on. So now to understand this process, we are going to take an example of a unicellular organism known as yeast, right? So yeast is a unicellular organism and can be seen only with the help of the microscope. So to perform this activity, we are going to need a microscope and we are going to need yeast, right? This is easily found in the market, right? So also we are going to if you might be thinking that for propagation or reproduction it might take time, but yeast, when it finds proper nutrients, proper temperature, optimum nutrients and optimum temperature, that is friendly environment, a proper environment, the yeast multiplies quite easily, it grows quite quickly, right? So we are going to perform an activity to understand the reproduction by the yeast, right? This activity should be demonstrated by a teacher so that it is easier for the students to focus on observation of what they are seeing. And you should also keep a copy and pen with you so that whatever you observe through the microscope you can draw. So what you are going to do is you are going to take a piece of yeast cake or a yeast powder. Both of these are available easily. Yeast powder is quite easily available on your shops. And what you are going to do is you are going to place it in some water, right? So you are going to add water in it and you are going to shake it well, and you are going to add a spoonful of sugar. Sugar, right? Sugar will act as the food material for the yeast, right? And water will provide the much needed moisture, right? Now what you're going to do is you're going to keep this whole setup in the warm area. Now when you keep it in the warm area, this provides the optimum temperature. So both the requirements, that is the food and the optimum temperature, is fulfilled, and what will happen, the yeast will start growing and it will start reproducing. So for observing this, what you're going to take is you're going to take a drop of solution from this whole this whole setup, the whole solution that you've made. You're going to put it on a glass slide and observe it under the microscope. What you're going to observe, please draw a diagram. Usually what you're going to see is this kind of structure where you will see an yeast cell. Few of the cells you might observe like this. Some of the cells might have this projection, like outgrowth in their body. This is called as the bud, right? So this is the developing bud and this is the new bud. Now what is the difference between the two? Over here you can see the nucleus is elongated, whereas in the new bud the nucleus has divided and it has given two nucleus. First is in new bud and the other is in your old parent body. Then what happens is that this develops in such a way that there is a boundary and the new bud is detached or separated from the parent body. Some of the buds might go off or they might detach from the parent body. Others might further grow and this would start forming buds of their own. So this is something which you will observe. Please draw the diagram of whatever type of yeast that you are observing in that liquid, right? In that drop of liquid, right? Now, this what you observed is the process known as budding, and this is the process that this unicellular organism, that is yeast, uses to reproduce. Let's understand it in a little better way, in a little detailed way. Now, what happens when the yeast is completely grown? This is the parent cell, it has a nucleus. Then what happens is, once it is completely grown, it starts forming a bud in this way, where as you can see the nucleus is elongated. Why it is elongating? So that it can be divided into two parts. One part of the nucleus will remain with the parent and other will go into the new bud that is being formed. Then further the cytoplasm also starts dividing, right? So this new bulb-like structure which is formed, this bulb-like projection which is formed, is called as the bud, right? Then you will see that over here this kind of construction starts coming, and this is the process of cytoplasm being divided. Once the cytoplasm is divided, this new bud is formed, right? This bud is the new yeast cell, right? So this is the new yeast cell that is formed. Many a times this yeast cell will dissociate from the parent cell and it will grow on its own. Sometimes it grows while it is attached to the parent cell and it keeps on forming more and more buds, forming the chain of yeast cells like this, right? So these are the daughter yeast that are formed, and this is the chain of yeast you might observe, wherein the largest cell that you will be able to observe will be of the parent yeast, right? So like I said that the yeast, if it finds optimum temperature, it finds favorable condition like optimum temperature and the food or nutrition that we added in the form of sugar, then the growth and the reproduction starts quickly, right? And this is so quick that many a times that before detaching only, these new bud starts forming smaller buds, right? So this process where the budding of an organism takes place, or where the formation of bud takes place, and from that bud the new organism is formed, is known as the process of budding, right? Now, another process that we are going to talk about is fragmentation. Now, you must have observed there might be a lake or a pond near you, and suppose this is the pond that is near you, and over here you found there was a small patch of green. After few days, you will see that this patch might have grown in such a way that the whole pond surface would appear greenish to you. How does this happen in very few days, or in a very little time? How does this small patch grows into full, it grows in such a way that it has covered the whole pond? This green slime, or the slimy green patches that you are seeing in the this stagnant body, is called as the algae, right? So usually you will find such kind of growth in the stagnant water. In rivers, such growths are not seen because the water is flowing continuously. So what happens exactly? Let's understand that whenever the algae finds proper nutrients and proper environment for the growth, right? So again, like I said, for any growth and reproduction process, optimum temperature and optimum food requirement is necessary, right? So favorable conditions are very, very important. So in this case, what happens is when the algae finds proper environment, what happens is that this algae will break into two parts, right? And it is a very rapid process wherein it breaks into two or more parts, and each parts develop into furthermore, each part develops into more of an algae. Then again it breaks into another two or more fragments, and each fragment will start forming new algae. It will start growing, and this is quite a rapid method, right? So this fragmentation is a quick method where you will find, because of this method, in a very short period of time the whole pond will be covered with the green algae. The example that we are taking over here is of the organism called as the Spirogyra. So this is a long ribbon-like algae wherein this is once it grows to a proper size, it breaks into two or say more parts, right? So it breaks into two or more parts, and then this further breaks into two or more parts. So you can imagine how much this growth is taking place, and it's quite rapid. That means it occurs quite quickly. That is why in very less time we see that the whole pond will be covered with the green algae. Right? This process where the parent body breaks into fragments to grow into a new plant is called as the fragmentation. Right? Now, once we've understood how the fragmentation takes place, let's understand another very important mode of asexual reproduction, and that is spore formation. Now, you've already learned in chapter 1 that the fungi, right, that grows on the bread piece, fungi grows on the bread piece when the bread piece is kept out in the warm and humid environment.
To show the growth of fungi. Materials required: moist bread, petri dish, microscope. Method: to observe bread mold, place moist bread piece in a petri dish. After a few days, examine the growth of microorganisms on the bread piece under a microscope. You observe a fungus rhizopus under the microscope that is commonly called bread mold.
So you might wonder that from where do these fungi actually come on that bread piece, right? So fungi grows through the spores that are present in the atmosphere. These are the spores that are produced by the fungi, and these keep floating in the air, and as soon as they find optimum temperature, or rather I should say favorable environment, which includes optimum temperature, food resource, these spores settle down and start forming new fungi, right? So in this way, the spores are able to travel a long distance. These are light and they are able to, they are able to float in the air for very long distances. If you observe the fungi that grows on the bread piece, you will be able to observe these spores which are present, which looks these spores which are present, and these spores are the ones which further helps in the formation of fungi. These structures are called as the hypha, right? So this is something which you can observe with the help of a microscope. Now, spores are basically asexual. Spores are basically asexual reproductive body. Please understand, it is a very important point because many a times students get confused with spores and seeds. The reason is that both of the spores and seeds remain as is until they find favorable environment. But the difference between the two is that seeds are sexual reproductive bodies, whereas your spores are the asexual reproductive body. So each spore is covered with hard protective covering, so that when they are floating in the environment, they are safe, right? Until they find favorable environment, they will not germinate, or they will not form a new organism, right? So what are the favorable environment, or rather I should say what are the unfavorable environment which the spores need protecting from? It is high temperature and low humidity. That means hot and dry environment is dangerous for these spores, right? But in the case of favorable environments, that is optimum temperature and high humidity, these spores settle on things like bread pieces, leather pieces, and they start forming the new organism. Apart from fungi, there are ferns which also reproduce by spore formation. Ferns are students, the plants which are used for decorative purposes, right? All of you must have seen ferns which are available on maybe it is present in your schools or even in your societies or colonies or your home. These are decorative plants. So if you look at the leaf, suppose this is the leaf surface which is visible to you. When you look at it and look at the backward side, you will find these small, you will find structures which are sori. These are called as sori, and these are spore forming bodies, right? So these are spore forming bodies, and through these soris only we see that the spore formation takes place. So the plants like moss and fern also reproduce by spore formation. Apart from that, we also observe fungi which is using the method of spore formation for the reproduction purposes, right? So this is all about the asexual reproduction, in which we saw the vegetative reproduction, the process of budding, fragmentation and spore formation. I hope you were able to understand the whole process properly. Now let's answer some questions.